actually, now that I just moved to Australia, much closer to Korea. So I hope to visit soon as the pandemic slows down. So today, as you heard, I'm going to be talking about extracellular vesicles for both therapeutic and diagnostic applications. But before I do that, I just want to share some beautiful pictures I took of Korea in 2018. And here we see Seoul as well, and had some wonderful food and even visited Bio. So you have some really fantastic extracellular vesicle companies, but also, of course, stellar extracellular vesicle research in your institutions and universities. So it's a pleasure to be invited to speak here today. And thank you also to KDOC for organizing this. And so you've heard from the great speakers in the morning session about what extracellular vesicles are, and many of you work in the field, but I just want to give a quick overview sort of from my perspective. And so what really characterizes these extracellular vesicles is that they're on the nano scale, so approximately 150 nanometers. And here you see one of our cryogenic transmission electron microscopy images, and you see this characteristic lipid bilayer. And so these extracellular vesicles contain various biomolecules such as RNA, proteins, and glycans. And depending on their biogenesis, they can be called microvesicles or exosomes. And the microvesicles, as you know, bud from the cell membrane, while these exosomes come from multivesicular bodies. But today, we don't really have good enough isolation methods or characterization methods to really distinguish whether we're working with microvesicles or exosomes, which is why this general term of extracellular vesicles is preferred, and it will be the term that I will use throughout this presentation. And so all the cells that have been studied have been shown to release these extracellular vesicles, and they've been shown to play an important role in cell communication, both over short distances, so between cells in the same tissue, as well as over long distances, so from one organ to another. And already in 1946, if we look at sort of from a historical perspective, there was a paper using high-speed centrifugation showing that there was something in the plasma in that nano size range that had this clotting ability. And so at that moment, they of course did not know it was extracellular vesicles, but later we know that they were actually looking at extracellular vesicles. And then in the 1960s, they were first given names such as membranous structures, little particles, or virus-like particles, and platelet dust. And then in 1971, the word extracellular vesicle was used for the first time in the literature, I think in the title of a manuscript. And then in 1978, they were shown to carry bioactive molecules. So in this case, enzymes. And then in 1981, the term exosome was first introduced to refer to these extracellular vesicles. And actually, the first speaker in the morning session has written a great paper sort of looking at the terminology historically. So I recommend you to look into Ken's paper in the Journal of Extracellular Vesicles talking about the terminology. And then in 1998, some important mouse studies were performed, and some of those were then used to do the first clinical studies in that involved mammalian extracellular vesicles. So as you heard, Ken mentioned that already for 20 years, extracellular vesicles have been used clinically in certain countries, but those are from bacteria. So we don't yet have any mammalian clinically approved extracellular vesicles. So perhaps one of you in the audience will be the first to bring that to market. And so we know that these extracellular vesicles have important roles in cell communication. So you could imagine that there would be very promising sort of therapeutic and diagnostic applications. And so why don't we yet have any clinically approved mammalian extracellular vesicles. And I think the main challenge, as you know, is really producing these extracellular vesicles, so concentrating them and purifying them. And the most common method in the literature is ultracentrifugation, but as you know, it results in very low quantity of extracellular vesicles, low purity, because you end up co-pelleting a lot of small molecules like free proteins. It's time consuming, it's labor intensive, and in most cases, it's not really clinically scalable because GMP facilities don't really have this ultracentrifugation capacity. And even if they did, there's still a limit in terms of what volumes can be used. And then it also causes damage. So this is the flip-flap railway, which is the roller coaster with the highest G-force in history. And so I think it's about 12 G. So you can imagine these centrifugation speeds that we are exposing these extracellular vesicles are much, much higher. So they will release their content, they'll aggregate, and the membrane is going to become damaged. And another challenge that we really have with these extracellular vesicles is that, in between the centrifugation steps, you have pipetting steps. And so this introduces human error, and you have low batch to batch consistency. And we know that this is one of the main things that regulatory agencies look at, is that we can produce similar batches. And so what my lab and also many other labs across the world, as well as Exaco Bio have been focusing on, is to find alternative isolation methods to overcome these challenges. So one of these methods is tangential flow filtration, and here's just a short video showing the liquid going through this tubing that is attached to filters in this commercial equipment. And of course, this commercial equipment is usually sold for protein purification, but if you change the parameters and the filters, you can effectively isolate extracellular vesicles. And my lab typically does this in 2 steps. So in the first step, we use a filter where the liquid flows tangentially, and this filter has pores that are 600 nanometers. So we get rid of anything that is above 600 nanometers. So for instance, cell fragments. And then in the second step, we use a smaller filter around 30 nanometers. So we get rid of everything that is below 30 nanometers, so free proteins and so on. And the advantage of this tangential flow filtration is that you don't get as much pore clogging because you're not putting pressure directly on the filter. And so then we use nanoparticle tracking analysis, like most labs do for the analysis. And of course, the software here is able to analyze this video and tell us the size distribution and concentration. So back in 2018, Dr. Busato, a postdoc in the lab, she compared ultracentrifugation, the most common method for extracellular vesicle isolation, to our method of tangential flow filtration with the same starting material. And what she found was that the yield was substantially improved with tangential flow filtration, but also that the purity was improved. So here on the y-axis, you see albumin, and albumin is a common contaminant found in the blood. And so with ultracentrifugation, you end up co-pelleting a lot of that protein while you get rid of most of it with tangential flow filtration. What you also see is that the error bars are very large with ultracentrifugation. So depending on the batch, you get different levels of purity. And so then we can also look at the size distribution profiles. And when we do ultracentrifugation, different batches, fall all over the place here, the dashed lines. But with TFF, the size distribution is more similar. And then we, of course, look at EV-enriched markers such as CD63 and CD81 and see that they're present, and the absence of intracellular vesicle markers such as calnexin. But cell culture medium is a pretty simple solution. So we also wanted to see whether we could use patient-derived liquids, so liquid fat such as lipoaspirate, and put it through tangential flow filtration. And we did see good batch-to-batch consistency. And again, we were able to isolate extracellular vesicles with those EV-enriched markers and lack of intracellular markers such as calnexin. You do see a little bit of calnexin here, and that is probably because as the surgeon is doing the liposuction, some of the cells rupture, releasing those intracellular vesicles. And then we also looked at sterility and yield, looking at different tests such as mycoplasma, endotoxin, bacterial growth for 14 days. And we're able to show that we don't introduce contaminants from the moment the sample is, for example, taken from a patient through the tangential flow filtration system until we get the final extracellular vesicles. And what's really remarkable is that if we take 50 milliliters of lipoaspirate, so we all have 50 milliliters of sort of liquid fat in our bodies, we actually get enough extracellular vesicles to inject 50 mice at a very high concentration. And so, of course, one of the challenges with using cell culture is that we don't get enough extracellular vesicles. And we've also done many studies focusing on storage. And again, the most common method to store these extracellular vesicles is in PBS. But we did notice that there are some fluctuations in the concentration when we store in PBS. But when we use the biocompatible cryoprotectants such as sucrose, the vesicles seem to be, the properties seem to be preserved. And of course, we know that many synthetic nanoparticles, as well as, for example, the COVID-19 vaccines, they are also stored in sucrose. So it makes sense that if you want to work with extracellular vesicles, there should be some type of cryoprotectant. And in the case of PBS, we see that sort of the concentration and the size is changing. And that could be due to various reasons, but for instance, degradation of extracellular vesicles. We also looked at some markers, and we found that if we look at the surface markers, they are enriched in the extracellular vesicles stored in sucrose, potentially suggesting that some of these proteins on the surface are removed when you have ice crystal formation in PBS. But what was even more interesting is that when we looked at cryogenic transmission electron microscopy in collaboration with the Talmon lab in Israel, what we found was that the extracellular vesicles that were preserved or stored in -80 in sucrose, they frequently had these molecular protrusions on the surface, while the extracellular vesicles that were stored in -80 in PBS had a smooth surface more frequently. So potentially this indicates that if you just store extracellular vesicles in PBS, you could be stripping some of the important surface molecules. And so we are excited to look further into this to see what type of surface changes are happening if we don't use a cryoprotectant. And so now that my lab had tried to overcome some of the main challenges in terms of isolation and storage, I'll share some of the applications that we focused on. And one is using these fat or adipose extracellular vesicles to treat inflammation. And the other is focusing more on diagnostics in terms of cancer metastasis and looking at the sugars or the glycans, because most labs have focused on extracellular vesicle RNA and proteins as biomarkers, while the glycans have been ignored for the most part. And so I'll start by talking a little bit about the therapeutic indication. And here we've teamed up with several investigators, but one of them is Dr. Fairweather at Mayo Clinic in Florida, and she has spent decades optimizing a myocarditis mouse model. And as you know, myocarditis is inflammation of the heart muscle, and it's responsible for 20% of all sudden deaths in the US. And the most common reason used to be Coxsackie virus B3. Today, it's probably COVID-19. But myocarditis can be caused by viruses or also other reasons. But in this mouse model, it's a hybrid model with a low viral load, and you mix it with hard protein. So it's an autoimmune viral model where you can see that infection peaks at day 14, and this inflammation can then be assessed, for example, through histology. And so what we did here is that we took, instead of taking the adipose-derived mesenchymal stem cells that have been published in hundreds of papers showing that they have some type of anti-inflammatory effects through the release of these extracellular vesicles, we decided to directly use the adipose tissue, and this was because using cell culture is quite a slow process. So it takes months to expand these cells, especially if you want those mesenchymal stromal cells. During this expansion process, it becomes expensive, especially if you're using it for clinical applications. We all know that if we put cells on 2D plastic, we may lose important properties. This is not really their native environment. Also, in most cases, we use exogenous agents that we add to cell culture, such as FBS, and regulatory agencies, they prefer not to have products from other species, so like cow proteins and so on. So that's another challenge you have to think about if using cell-derived extracellular vesicles. And again, you get very low yield despite using improved methods such as tangential flow filtration. So we asked that perhaps if we took healthy individuals, healthy donors, and took some of their adipose extracellular vesicles, could they potentially have similar anti-inflammatory properties as those adipose-derived mesenchymal stromal cell extracellular vesicles described many times in the literature? And so this is Lipogem's device. It's FDA approved, so clinically approved in the United States, developed by Italian surgeons. And basically, you shake the fat in this device, and it will separate the starting product into cells, concentrated cells, and then a sterile waste bag, which is the acellular fraction. And this sterile waste bag is usually thrown away on a regular basis in the clinic because the cells are used for clinical procedures such as injection for sport injuries, so injection into the shoulder. And so what we wanted to do is actually take these waste bags that were not being clinically used and run them through the TFF as a potential therapeutic modality to then get those adipose tissue extracellular vesicles. But at the same time, we wanted to compare donor-matched MSC-derived extracellular vesicles. So we grow them for a month, we make sure they have those mesenchymal stromal cell markers, and then we take the conditioned cell culture medium and we run it through TFF, and we obtain those sort of conventional cell-derived extracellular vesicles, or cell culture-derived. And so the upper process will take months, the lower process will take just a few hours. And when we compare the conventional to the new method, we can see that the process that is very quick, just a few hours, will result in much more extracellular vesicles than the process that takes several months. So in terms of yield, we definitely have here an advantage. But of course, it's important to see whether they have any therapeutic benefit. And so one of the sort of simple things we've done before looking at this myocarditis model that I mentioned was just look at macrophages, both primary macrophages from humans as well as various other macrophage cell lines. And the reason we do this is because these macrophages are known to infiltrate into the heart during myocarditis, where they activate then the toll-like receptor 4 pathway and release inflammatory cytokines. And so a simple model is to add LPS, which is a TLR4 ligand, and then we can add these adipose tissue-derived extracellular vesicles into the culture and look at inflammatory cytokines. And surprisingly, we saw that the adipose tissue extracellular vesicles could suppress inflammatory cytokines to a very similar extent as the donor-matched extracellular vesicles from the cell culture from MSCs, which are not shown here, but also to a similar extent as has been shown in the literature many times. So surprisingly, they do have similar anti-inflammatory properties in this simple cell culture model. And so we started to wonder what could actually be causing this sort of suppression of TLR4 induced cytokines. And so we looked at the most abundant microRNAs that were present in these adipose tissue extracellular vesicles, and we identified a few that had already been linked to TLR4 suppression. But when doing further analysis, we found that especially microRNA 451A was able to mimic some of the effects of the extracellular vesicles. So it seems that potentially this microRNA could be partially responsible for the suppression of TLR4 cytokines. And then we can also, what we also did is add this inhibitors of microRNA 451A together with the adipose tissue extracellular vesicles, and we saw that their effect is then minimized. But of course, we know that there's so many things in these extracellular vesicles, so it's unlikely to be one biomolecule that is responsible, but instead a mixture of biomolecules such as proteins, microRNAs, glycans, and lipids that together then cause some type of anti-inflammatory effect. And in addition to macrophages in cell culture, we've also looked at endothelial cells, because we know that in myocarditis, those endothelial cells will recruit macrophages. And in this case, we can use TNF alpha to induce this inflammatory phenotype and then add the adipose tissue extracellular vesicles. And again, see that there is some suppression of these adhesion molecules linked to inflammatory pathways. And then we injected these adipose tissue extracellular vesicles in the mouse model, and through histology, we could see that inflammation was reduced compared to the control mice. We could also see a reduction in TLR4 in the heart. We saw less infiltration of immune cells, such as macrophages, and we also saw some beneficial effects on the complement system. So we're excited to further look at these in vivo studies to understand mechanistically what could be going on. But in addition to myocarditis, we've also teamed up with Dr. Hickson, who's at Mayo Clinic, Florida, to treat diabetic kidney disease. And it's the most prevalent cause of kidney disease in the United States. And it's expected to increase even further because the population is becoming older and more obese. And so she has developed various models to look at diabetic kidney disease. And we can see that when we add or give these mice extracellular vesicles, we have improved functional parameters and less fibrosis. But what we also see is that we have reduced macrophage infiltration and also some interesting effects on senescence that we're looking further into. But then we asked, could we go one step sort of further to improve the therapeutic benefits? Could we load these adipose tissue extracellular vesicles with drugs for combination therapy? So to exploit both the intrinsic anti-inflammatory properties as well as, using conventional drugs, for example, to boost this effect. And so there's several strategies that can be used to load drugs into extracellular vesicles post-isolation. So in this case, because we're taking them directly from patients, we're not using any type of cell culture. We can't utilize genetic engineering, but instead we can use physical and chemical methods to get these extracellular vesicles to get the drugs into the extracellular vesicles. And this includes sort of electrical energy, sound energy, freeze-thaw cycles, mixing, and extrusion. And the most common method, or the most simple method to do this, is actually mixing. And this only works for hydrophobic drugs that can go into the lipid bilayer. And so we use guanabans, which is a clinically approved drug that is shown to inhibit the TLR4 pathway, to see if we could further suppress these inflammatory cytokines. And we simulated in vivo conditions, so body temperature, mixing, and the presence of serum proteins. And we could see that after this drug was loaded in the lipid bilayer, it was slowly released over time. And when we looked at the combination, we saw that these inflammatory cytokines were further suppressed when we had both the extracellular vesicles as well as the drug loaded inside. But in addition to, clinically approved small molecules, we can also explore loading more complex therapeutic agents into these extracellular vesicles. And in this case, we use a hybrid approach. So we merge synthetic nanoparticles with extracellular vesicles to enable loading of, for example, proteins like Cas9 or RNA. And in this case, if you look at step 4, size exclusion chromatography can be used to then separate the synthetic nanoparticles and the unloaded protein from the protein that was loaded inside. And I'll show you some of the results here. So we loaded Cas9, which of course is a large protein, into the extracellular vesicles using this hybrid approach of cationic nanoparticles that were complex with Cas9 and then complex with extracellular vesicles. But you can see that the extracellular vesicle enriched markers were preserved after this hybrid system was created, and we can also see that Cas9 only shows up in the extracellular vesicle hybrid group, and we get pretty high loading efficiencies in this case. And one thing that is good to always look at is the zeta potential, because it's an approximate measurement of the surface charge, and the surface charge has been linked to toxicity. So as we know through many preclinical and clinical trials, if you have a cationic, so positively charged nanoparticle, you will elicit some type of toxicity. And so even though these cationic nanoparticles are very good at loading RNA and proteins, they are also toxic due to their cationic charge. But when we create this hybrid system in red, we can see that the zeta potential is still similar to the native EV, so it's still negative. But even so, we get, when we look at how much Cas9 can be delivered into the cells, we see that actually the EVs outperform electroporation. Lipofectamine is still, the best option, but again, lipofectamine is not feasible for in vivo use due to toxicity. And so when we look at the toxicity in a very simple cell viability assay, we see that when we have the synthetic nanoparticles here, termed pulsine cationic with Cas9, we see a reduction in cell viability. But when we had the hybrid system with the EVs, we see less cytotoxicity. And so these conventional approaches such as electroporation and cationic nanoparticles can be effective, but they also cause cell death. And extracellular vesicles could be an emerging strategy to prevent cell death while still being able to deliver RNA and proteins into cells. And so that was a brief overview of what we've sort of been working on the therapeutic side. But then on the diagnostic side, we've been looking at these glycans, like I mentioned. And the reason we've been focusing on cancer metastasis is because, as you know, 90% of cancer deaths are due to metastatic disease. And so one of the problems is that we can't detect the early stages of metastatic disease. We usually catch it too late. And we know that these extracellular vesicles that are released from the primary tumor, they will go into the blood and go to distant organs, and they are contributing to the formation of the pre-metastatic niche. So these cancer cell-derived extracellular vesicles are being released at a very early stage of the metastatic cascade. So it makes sense that potentially these extracellular vesicles could then be used as biomarkers to detect early stages of metastatic spread. And many labs have done so, looking at RNA and protein cargo, but the glycans have been ignored due to technical difficulties in high throughput analysis. And this is a shame, because we know that glycans are really involved in cancer progression, especially, adhesion between cells and membranes. And so you could imagine that these glycans would play a role in extracellular vesicle-induced pre-metastatic niche formation. And so in this case, we've teamed up with Dr. Borges at Arizona State University. And for diagnostic purposes, we don't use TFF because TFF is optimized for large volumes, hundreds of liters. And so for the diagnostic purposes, we want to be able to use very small volumes. So we use size exclusion chromatography, and we combine this with glycan node analysis, which is sort of a new way of rapidly looking at all the glycan classes on extracellular vesicles. And so we can take parental cell lines. So this is triple negative breast cancer from humans, a very well-known cell line. And when it's injected into mice, and you go through several cycles where one of 20 mice has a brain metastasis, you then resect those cells and inject it in another cohort of mice. And if you repeat this cycle, you'll actually be able to get a very brain-tropic cell line that is otherwise quite similar to the parental cell line, except that it goes to the brain. And studies have shown, for example, already in Nature 2009, that even though these metastatic cell lines have been created in a mouse model, they still have very similar gene expression to patients with breast cancer brain metastasis. So there is some clinical relevance to these cell lines, but it also makes sense to compare them side by side, because the one difference here is that they go to the brain. And when we took the extracellular vesicles from the regular cancer cell line and the one that metastasizes to the brain, we could see many differences in the glycan expression. And then we can, based on these glycan nodes, in many cases, it will actually tell us, we can predict which specific glycans are enriched. And we can use ELISA then to confirm. So in this case, we've been able to identify certain glycans that are enriched on extracellular vesicles from breast cancer cells that go to the brain. And we can then use enzymatic stripping to remove these glycans from the surface. And in addition to assessing their role as diagnostics, we can also look at the mechanistic role. So one of the things we found is that, you know, these extracellular vesicles also have different interactions with immune cells. And this could also potentially be mediated by the glycans that are on the surface. And so other things we've found is that the extracellular vesicles that go to the brain, they actually interact more with LDL and cause some type of aggregation. And so we're very interested in exploring in general, you know, what is the role of extracellular vesicle lipoprotein interactions, and could those interactions be mediated by glycans, and do they have a pathophysiological role? And so we've done, this is AFM, but we've done also, we have some very exciting cryogenic transmission electron microscopy showing that extracellular vesicles actually interact and fuse with lipoproteins in vivo conditions. But we don't know what that means yet. But then this is just an example of looking at human circulating extracellular vesicles in the plasma, and for the first time sort of characterizing what are the extracellular vesicle glycans in healthy patients, in healthy plasma. And when we compare, you know, crude plasma and the extracellular vesicle fraction from plasma, we see that the glycans are very different. So we see that extracellular vesicles have specific enrichment in certain glycans. So, you know, today we use the Western blot, so we use proteins to authenticate our sample, but in the future, maybe glycans could also be used for sort of authentication of whether we have an EV sample or not. But we don't know whether that's the case yet. So now we're looking, at this moment, we're looking at a large cohort of breast cancer metastasis patients, and we're looking at the circulating EVs and the glycans on the surface. And so hopefully in the next few months, we'll have some exciting results to report of whether some of those glycans that we identified in our in vivo models and in our cell culture models are also visible in the plasma, and whether those general patterns are different between patients that don't progress to metastatic disease and patients that do, for example, brain metastasis. But what's also interesting to note that if we only take 3 healthy donors and we isolate their EVs from the plasma, the glycan profiles are extremely similar. And this is a good indication that we could potentially detect any variation if the patient had, for example, cancer, because there's little variability in the healthy population. And so we're looking forward to getting those results. So in conclusion, sort of the main question we've asked is, can we tap into the body's text message system to improve healthcare? And so these extracellular vesicles are the text message system of the body. And we think we can do that both for therapeutic applications, specifically focusing on adipose-derived extracellular vesicles because of their scalability and looking at inflammatory diseases such as myocarditis and diabetic kidney disease, but also looking at dual role. So not only their intrinsic anti-inflammatory properties, but also their use for drug delivery. And this enables various combination therapy approaches. And then on the diagnostic side, can we really tap into these extracellular vesicle glycans that have been largely ignored, and especially for metastasis, because we know this happens early in the metastatic cascade, that some extracellular vesicles are released from the primary tumor. So with that being said, I really want to thank all the lab members that contributed to this fantastic result, and they worked very hard in the lab. Our collaborators, especially Dr. Talmon in Israel, who's a cryo-TM expert, Dr. Fairweather and Hickson, who have inflammatory animal models, and our collaborators, Dr. Borges, who has this new glycanode throughput analysis model, and then our funding sources. And you can follow our scientific updates on Twitter at Wolfram Joy. And if you're interested in coming to Australia to work in the lab, please send me an email. Thank you.
Thank you, Joy. It's an excellent presentation. We have a couple of questions from the audience. The first one is, how can you measure the purity of exosomes?
Yeah, this is an excellent question. So
okay.
I think there's no clear method of doing that, but sometimes you can compare the protein to lipid ratio. So I think that is very valuable. So, or you can compare, the RNA to lipid ratio and so on. But overall, since the extracellular vesicles vary so much, it's difficult to say that, one specific thing. For example, we used to say that apolipoproteins, they indicate that extracellular vesicles are not pure, but recent results indicate that apolipoproteins are actually part of the extracellular vesicle protein corona. And now that the lipoproteins may actually be fused to the extracellular vesicles. So that means that it's not necessarily that they're impure, but that's just how they're, biologically active by forming these complexes.
Okay. The second question is, do you think it is possible for exosomes to enter clinical trials by only purifying through TFF?
It's a good question. I guess it depends on what the source is. And as Ken mentioned, is that, some of those impurities may actually be neutral. So they may not cause any bad effects, or they may even be beneficial. So depending on the application, depending on the source, it may not be necessary to purify further than TFF, or you may actually be doing a disservice by purifying further by removing some of the biological activity.
Okay.
Yeah.
And I think this is lipo nanoparticles have lower anti-inflammatory properties compared to extracellular vesicles. Do you think lipo nanoparticles have more therapeutic benefits compared to extracellular vesicles? Do you understand the question?
Yeah, I think either it could mean synthetic lipid nanoparticles, or it could mean, for example, HDL. But because there's so many different synthetic lipid nanoparticles, I think I'm going to understand it as HDL. So I think HDL has been shown to have anti-inflammatory properties. There are clinical trials being done with HDL therapeutics with 17,000 patients enrolled. So I think the lipoprotein field, lipoprotein therapeutics, are ahead of the extracellular vesicle field in terms of scale up. So they're already doing, enrolling 17,000, huge clinical trials with HDL. So but then again, it's difficult to compare because, extracellular vesicles, you could have extracellular vesicles that are very pro-inflammatory. You could have extracellular vesicles that are anti-inflammatory. So there's such a big range. So some of them may be superior to HDL, some of them may have similar effects to HDL, and some of them may be worse.
And if GBG, I think it's a chemical, can be easily loaded into AD, adipose tissue-derived extracellular vesicles due to hydrophobicity, does it mean GBG can also leave ADEV easily? So easy loading, easy
that's
release.
That's a great question. So when we did the release with serum, it did not leave. As you saw, I mean, it slowly was released, but that's of course not really mimicking the in vivo conditions. So in vivo, if there's more cell membranes to interact with, it could be released. But I think it needs something else that is hydrophobic to jump from the extracellular vesicle membrane to another lipophilic structure.
And here a question. I am curious about the size range of proteins that can be loaded into exosomes.
Amazing question. So in that paper, we looked at 3 different size proteins. We actually loaded albumin, then we loaded Cas9, and then we made a Cas9 conjugate that was even larger. And so all 3 we could load, but the bigger the protein was, the lower the loading efficiency.
Okay, a few more questions here. I think it is very fascinating to isolate exosome from acellular portion. Do you think it is possible to isolate exosomes from other tissues?
Fantastic question. So I think there's some groups looking at, there's a lot of groups looking at plasma, but of course plasma has every single extracellular vesicle. But for example, placenta and other tissues may have unique properties. So I do think it's possible. But if it's more a solid tissue, then you have to think about enzymatic digestion or how you're gonna process it before the extracellular vesicle isolation.
Okay, I also have a question about the stability of exosomes or lipoparticles from the adipose tissue. You stored exosomes in the -80 in the presence of cryoprotector, and you said it's quite stable. So have you tried to lyophilize exosomes for keep them in a very intact form?
Yeah, we have not. I have not, but I know some groups, for example, in Mayo Clinic, Minnesota, they do lyophilize, as well as there's some groups, Professor Pastorin in Singapore, she has lyophilized them and got pretty good bioactivity. So I do think it's possible, but we haven't tried that yet.
Okay. And it's very interesting to see that the adipose tissue-derived lipo or nanoparticle has some anti-inflammatory effect. But I wonder, the adipose tissue, particularly the adipose tissue in the obese patient, they may have a inflammatory process in the adipose tissue. So, do you see the difference? Any difference from the normal adipose tissue and obese adipose tissue?
Yeah.
Do you understand my question? Okay.
Yes, this is a beautiful question, and it's a question that we get asked. We've been asked before, not only obese but also patients with, for example, diabetes, so with metabolic disorders. And so I do think you probably would see an opposite effect, because as you said, many studies show that in obesity or in diabetes, the adipose tissue actually has, for example, macrophages that are pro-inflammatory. So we would love to, in the future, compare the healthy adipose tissue vesicles to disease obese vesicles, and perhaps get the opposite effect. And so I think it's important to make sure that you don't have an adipose tissue that is, obese or metabolically abnormal.
Okay. So the final question is, what do you think the most important active ingredient in adipose-derived nanoparticles to have the anti-inflammatory effects? Of course, it's quite complicated, but you have to do more experiments to identify which molecule is more critical. But do you have any data or any idea?
Yeah, so this microRNA we found that is very, very abundant, third most abundant microRNA. It does mimic some of the anti-inflammatory properties, but it's very difficult to compare the exact dose. And so potentially it could also be a very sort of general mechanism where the lipid bilayer will just bind to endotoxins. We don't know if it's something, with the surface being a sponge for inflammatory signals, or if there's specific microRNA, and most likely it could be a mixture of these specific effects and maybe just this sponge effects.
Yeah, I think we don't have question anymore, so we are a little bit ahead of schedule, but I think it's okay. So thank you, Joy. It's wonderful talk. So okay, I will introduce the next speaker. So Joy, don't leave. Stay.
Yes.
If you have a question, please have
Wonderful.
questions. Yeah, okay. And
thank you.
Thank you.